Welcome back to Unraveling Human Physiology, where we try to make physiology easy for you to understand. Hi, in this video we are going to talk about hematopoiesis and hematopoietic stem cell regulation. Blood is a very interesting tissue as blood cells are not embedded in a fixed matrix of proteins, but instead they are in a liquid form and circulating all around the body. And here's our first question. How long do blood cells last? Duration depends on the cell type. Red blood cells last some 120 days. White blood cells last some hours. They can last longer but not circulating in blood. And platelets last from 7 to 10 days. Then, how are new blood cells generated? New blood cells are generated in a process called hematopoiesis. Hematopoiesis is a complex process where from one common cell, the hematopoietic stem cells, the cell divides and differentiates to form the different kinds of cells in blood: platelets, red blood cells, macrophage, neutrophils, eosinophils, basophils, T lymphocytes, B lymphocytes, and natural killer. Let's start from the beginning, the hematopoietic stem cell. As the hematopoietic stem cell can differentiate to form any of the blood cell types, we said that it's a pluripotent cell. If we count all the cells in the bone marrow and we classify them, we will see that there are very few hematopoietic stem cells. Only 0.04% of the marrow cells are hematopoietic stem cells. Another characteristic is that they are indifferentiated and has embryonic characteristics. If you see them in a microscope, you won't be able to distinguish this cell from another cell. It doesn't have specific characteristic of blood cell types like red blood cells, for example. It's just a simple cell without characteristic. We need to perform biochemical tests just to know that it's a hematopoietic stem cell. Hematopoietic stem cells have a very high proliferative capacity. They can divide indefinitely, but not every one of them is really proliferating at the same time. Only 5% of hematopoietic stem cells are in active proliferation state. The rest of them are quiescent. We call these dormant cells G0 phase of the cell cycle, and now on I will call them G0. So this is a very highly regulated process, which is logic. Imagine we have an hematopoietic stem cells which can divide indefinitely and give rise to every blood cell type. If it's not a regulated process, it would be a disaster. We will have any number of cells and blood will not function well at all. So stay with me in this video and I will explain you how is this process regulated. Hematopoietic stem cells can divide in two ways. The third form of division is a proliferation that gives rise to an identical hematopoietic stem cells, and we call that self-renewal. The other form of proliferation is proliferation and differentiation. In this type of division, the hematopoietic stem cells is commissioned and give rise not to another hematopoietic stem cell, but instead to a progenitor, which can be a lymphoid progenitor or a myeloid progenitor. But what is a progenitor cell? Progenitor cells, or colony formation units, are cells that can give rise to a blood cell type line and all its precursors, for example, red blood cells and all its precursors, neutrophils and all its precursors. They are so multipotent cells. They can give rise to different kinds of cells, but only if they are line specific. For example, a red blood cell progenitor can give rise to red blood cells and all its precursors, but it won't be able to divide into lymphocytes. And there are cells that they are not differentiated, so if we look at them at the microscope, we won't be able to differentiate a progenitor from a hematopoietic stem cell from another type of cells. We need to perform some biochemical studies to be able to differentiate for example, progenitor from a hematopoietic stem cell. The capacity of progenitor cells to differentiate is dependent on the presence of colony-stimulating factors that are present in the media. They cannot self-renewal. That means that if a progenitor cells divide, it cannot form another identical progenitor cells, but it will be forming a differentiated form of cell. And they have also a very high proliferative capacity. But it is not as in hematopoietic stem cells that can divide and divide and divide, but progenitor cell proliferative capacity is short-lived. They can divide a lot in a very short time, and then they stop dividing. So from a single progenitor cell, we will have rapidly lots of cells that will differentiate into a single blood cell line. There are different kinds of progenitor cells, which are more or less differentiated. The less differentiated progenitor cells are the pluripotential progenitors. We have two pluripotential progenitors. The common myeloid progenitors that give rise to platelet, red blood cells, macrophages, eosinophils, basophils, and neutrophils. And the lymphoid common progenitor cell that give rise to lymphocyte types and natural killers. Then we have bipotential progenitors that give rise to two blood cell type lines. Of bipotential progenitor, we can mention the erythroid megakaryocyte progenitor that give rise to platelets and red blood cells, and the granulocyte monocyte progenitor that give rise to macrophages and neutrophils. And finally, we have the more differentiated progenitors that are the unipotential progenitors that can give rise to only one blood cell type line. Obviously, we will have one unipotential progenitor for each blood cell type line. So we will have the erythroid progenitor that will give rise to the red blood cells, the megakaryocyte progenitor that will give rise to platelets, the monocyte progenitor that will give rise to macrophages, the granulocyte progenitor that will give rise to neutrophils, the eosinophil progenitor, the basophil progenitor. And for the lymphoid line, we have the lymphocyte B progenitor and lymphocyte T progenitor. All these progenitors, when they divide and differentiate, they will give rise to precursor cells. Precursor cells are the very first cells in the origin of blood cell types that can be differentiated morphologically in a microscope. You will be able to see in a microscope different kinds of precursor cells, and you will be able to point out which one of these precursor cells are. They are the most abundant cells in the medulla, and we can find precursor cells in different stages of cell maturation. Some of them will proliferate and mature, and other ones will just mature into the final mature blood cell type. And finally, we have the mature blood cell types that will eventually go from the medulla to the blood. Now that we know how the process goes, where does it take place? In the first month of the embryo, hematopoiesis occurs in the yolk sac, and we call this phase myeloblastic. After the 5th week, hematopoiesis begins to occur in the liver and the spleen, and we call this phase hepatic phase. Finally, after the 4th month of the embryo, hematopoiesis starts occurring in the bone marrow. We call this phase myeloid phase, and it goes on in the bone marrow all along our life. I told you before that all hematopoietic process was highly regulated. How is hematopoiesis regulation? Hematopoietic regulation occurs through the bone marrow microenvironment. And what is the bone marrow microenvironment? It's a media that is formed from the interaction between hematopoietic cells, non-hematopoietic cells, the extracellular matrix formed from these cells, that is collagen fibers, reticulin, and adhesive proteins, and cytokines that are present in this media. Cytokines are substances that are produced in cells of, for example, the bone marrow, and that can be able to inhibit or stimulate not only proliferation, but also differentiation of the different types of cells that we can find in the bone marrow. Which are the non-hematopoietic cells that are influencing hematopoiesis? We have sympathetic adrenergic neurons, non-myelinating Schwann cells, some mesenchymal cells like adipocytes, niche and osteogenic osteoblasts, arteriolar and sinusoidal pericytes, T lymphocytes, and macrophages. We will find specific interaction in a particular site of bone marrow called the hematopoietic niche. Hematopoietic niche are specific anatomical sites within the medulla where hematopoietic stem cells can either remain quiescent in some of the hematopoietic niche, or instead in other types of hematopoietic niche, hematopoietic stem cells are commissioned, they are proliferating, and they are in active differentiation. They are restricted spaces. So, for one cell to enter a specific hematopoietic niche, another cell that is within the niche must exit. We have two types of hematopoietic niche. We have endosteal niche, where we have quiescent hematopoietic stem cells. So most hematopoietic stem cells in an endosteal niche is in G0 cell cycle phase. And then we have vascular niche, where hematopoietic stem cells are active. So we will mark that hematopoietic stem cells are in G1 phase of cell cycle. Endosteal niche are near the bone endosteum. Near the endosteum, we will find osteoblasts. In particular, we are interested in a certain type of osteoblasts called niche osteoblasts, and of course some osteoclasts. These sites have an irrigation through central arterioles. In association with central arterioles, we will find periarterioral pericytes. These pericytes are innervated by sympathetic nervous system fibers, which are covered by non-myelinating Schwann cells. Osteoclasts that are present in the endosteal niche will degrade bone and will free calcium to the media. So calcium concentration in the endosteal niche is high. On the other hand, we have the arterioles that are near the endosteal niche that have a small diameter and little flow. So oxygen concentration in the endosteal niche is very low. The high calcium concentration, in addition to a specific cytokine called CXCL12 or SDF1 alpha, attracts the hematopoietic stem cells to the niche. CXCL12 is secreted by the pericytes and is a cytokine that produces chemotaxis, that is, it attracts the hematopoietic stem cells to where it is in high concentration. It also makes that hematopoietic stem cells stay in the niche and is a survival factor for the hematopoietic stem cell. Hematopoietic stem cells stays in the endosteal niche not only through the action of CXCL12, but also through interaction between molecules in the membrane of hematopoietic stem cells and the pericyte, like through angiopoietin 1, vascular cell adhesion molecule 1, and stem cell factor, and also between interaction between the membrane of the hematopoietic cells and the membrane of the osteoblasts through caderings. What happened with the hematopoietic stem cells once in the endosteal niche? Well, remember the Schwann cells? Schwann cells secrete TGF-beta1, which is an inhibitor factor. TGF-beta1 plus the low concentration of oxygen I told you before there is in the endosteal niche makes the hematopoietic stem cells stay quiescent. So, hematopoietic stem cells in the endosteal niche are in G0 phase of cell cycle. This quiescent state is helped also by other factors that we find in the endosteal niche that are thrombopoietin that comes from the liver, and the fact that CXCL12 is in low concentration. Why do hematopoietic stem cells not stay forever in the endosteal niche? Well, during the day, the sympathetic fibers are activated and secrete norepinephrine to the pericytes. That makes that the concentration of CXCL12 will be lower, and then there is less chemotactic factor to maintain hematopoietic stem cells inside the endosteal niche. So hematopoietic stem cells leave the endosteal niche and they translocate to the vascular niche. Vascular niche are in the central part of the bone marrow, near the medullary sinusoids. Adjacent to the sinusoid, we will find perivascular pericytes, and another type of cell called CAR cells. Perivascular pericytes are also innervated by sympathetic nervous system fibers. In the vascular niche, not only pericytes secrete CXCL12, but also CAR cells, and CAR cells secrete large quantities of this cytokine. So concentration of CXCL12 in vascular niche is higher than in endosteal niche. Remember I told you that CXCL12 is a chemotactic cytokine. So hematopoietic stem cells will be attracted to the vascular niche thanks to this cytokine. In the vascular niche, we also find interaction between molecules in the pericyte membrane and the hematopoietic cell membrane, and between CAR cell membrane and the hematopoietic cell membrane. Remember, we have angiopoietin 1, PKAN1, and stem cell factor. One characteristic of the vascular niche is, as it is far from the activity of osteoclasts, it would have a very low concentration of calcium. Another characteristic is, as sinusoids have large diameter and large flow, the concentration of oxygen is very high. So we have low concentration of calcium and high concentration of oxygen. These concentrations, in addition to the high concentration of CXCL12 that in a high concentration activates the hematopoietic stem cells, makes that hematopoietic stem cells will go through a quiescent state to an active state. As I told you before, an active hematopoietic stem cells can go through two types of process. The first one is self-renewal, where identical hematopoietic stem cells are created and they can repopulate bone marrow, and a part of them will go back to the endosteal niche, stay quiescent, and be able to be active for another time. And the other process that they can go is to proliferate and commissionate to form the different blood cell lines. Hematopoiesis follows a circadian cycle. During the day, hematopoietic stem cells are favored to be in the vascular niche, active and generating the different cell lines, and we will see a migration of mature blood cell types to the blood. This process is favored with the presence of norepinephrine secreted by the sympathetic nervous system. And during the night, proliferation of stem cells is mainly involved in self-renewal, and most of them return then to the endosteal niche. So the quiescent state is favored in the night. We observe that there are few cells that migrate from the medulla to the blood, and this process is favored by the presence of melatonin. I hope you have understand all the hematopoietic process and its regulation, and you have enjoyed this video. If you like this video, please give me a like, subscribe to my channel, and activate your notification to know when my next video will be on. Thanks, bye!